Good day ma'am, good day everyone. So today I will show you that how to transfer bacteria from one petri dish to another. So the first step is to sanitize the area with isopropyl alcohol. Then you have to take the alcohol lamp which is used to burn and kill the microorganisms. Then transfer the alcohol lamp into the lamina box. Inoculating needle should be kept in the isopropyl alcohol to keep it sterile. So inoculating loop needs to be heated over the alcohol lamp at 45 degrees Celsius so that, and it should be until it's red hot. But the next step is to take petri dish when contains the bacteria culture and ensure that inoculating loop is not that hot. So you have to check the temperature on it. You just have to tap on the petri dish, tap on the end of the petri dish. You have to wait a little to cool down. If it's too hot, then you have to just tap on and you have to wait a little to cool down your inoculating loop. You just have to pick your desired bacteria culture, then desired bacteria colony, and give it a light tap, and then so you just have to pick your desired bacteria colony and give it a light tap, and then divide your petri dish into 4 imaginary quadrants, 1 2 3 4, and then streak the bacteria in 4 quadrants. Then you have to sterilize the inoculating loop over the alcohol lamp, which is your sterilize it, and then over the alcohol. Once it's red hot, you have to wait until to cool it down. Then from the first quadrant, streak it to the second quadrant, and you have to repeat the process of sterilism and streaking until the bacteria until you cover the 4 quadrants over here, 1 2 3 4, which are covered. The next step is to seal your petri dish once you're done, and keep it upside down like this. Keep it upside down and at a room temperature. So now I will talk about the importance of proper aseptic methods in microbiology. It protects the integrity of a culture. Aseptic method ensure that you only have desired bacteria colony. It eliminates other microorganism communities and prevents cross-contamination and provides efficient results. It also provides safety to the microbiologist, protecting them from harmful microorganisms. So now we'll talk about staining bacteria or fungi. So the first step, or the primary step, is to sterilize slides with isopropyl alcohol. Make sure if it's clean, and also it shouldn't be cloudy. Add tiny drop of water, and get your petri dish containing bacteria. Get a petri dish which is containing the bacteria, and transfer the desired bacteria to your slide. And then inoculating loop should be sterilized at 45 degrees Celsius, which is obvious, over the alcohol lamp, which is over here, over the alcohol lamp. Check the temperature of the inoculating loop, as it shouldn't be too hot. To check for the temperature, you have to tap at the end of the petri dish. If it sizzles, it means that it's still hot, so you have to wait until it cools down. Once done, tap on the desired colony. Okay, once done, you have to tap on the desired colony, and then transfer it to the slide by mixing it with a tiny drop of water on the slide. Then allow the slide to dry. Once it's dry, you just have to keep it over the lamp. The slide, and after then you can dent the propyl staining method. So now the first step is to add crystal violet dye to your slide. You have to completely wash your slide with crystal violet and wait for 30 seconds to 1 minute for the bacteria to properly absorb the crystal violet dye. After 30 seconds, wash your slide with distilled water before adding iodine. Then add iodine and wait for 30 seconds to wait for bacteria to absorb it. Then wash it with distilled water, adding or washing slide with ethyl alcohol, waiting for 30 seconds to wait for bacteria to absorb it with the distilled water. Add saffron in, then wait for 30 seconds to 1 minute, then wash slide with distilled water, then dry the slide and view it under the microscope. So now I will show you the staining method for fungi. Add a tiny drop of methyl blue to a sterilized slide. The staining method is quite complicated in fungi as it contains spores and hyphae. Get a strip of the tape and give desired colony a light tape, and then transfer it to the slide containing methyl blue. Get a cover slip if it's clean and also not cloudy. Cover your slide and then view it under the microscope. Now, to get the hyphae, you need inoculating needle, then sterilize it and check the temperature is right. Pick desired colony of fungi and give it a light tap to get the hyphae. Transfer it to the slide containing methylene blue and cover with cover slip again and view it under the microscope. So now you have to take some precautions to take care while doing the staining process of fungi, as it's complicated and it's dangerous. So make sure all the fans in the labs are off so that the spores don't bloom in the lab. So this is my video on how to transfer bacteria